Hey, Ava, I think we can start the lecture as others are joining in. Thank you.
Awesome. Sounds good. Hi, everyone. My name is Ava. I am a fourth year PhD student at the University of Washington. I'm actually a colleague of Ice who gave the lecture a couple weeks ago. He's actually the person who trained me in graduate school, which is pretty cool. So today we'll be talking about engineering bacterial therapeutics. And before we actually do that, there's a couple of course reminders for all of you. So yeah, welcome to week 6 of the course. It's very exciting. Just a reminder, homework 5 was due before this class, and group selection is happening now. So if you're still looking for a group, please let us know. There's a Google form that Heidi can send to you, or Emma can send to you. And if you've filled out the form already, groups will be assigned in the next couple of days. And you'll hear more about that, I think, from the other people from SynBio4All. And for the homework for this lecture, you also need to submit this Google form. So what we want is at least one member of your group to submit 2 project ideas to the form that Emma will be sending in the chat now. So just as a reminder, make sure that you fill out this form in addition to homework 6. Cool. And so with that, let's get into, if there's no questions, which Emma can be on the lookout for, we can go in and start with our lecture outline. So today, some of the things that we will talk about is first, we'll do a glossary. So we want to define some of the different terms. What are the differences between pre and probiotics, and what are living therapeutics? And then we'll talk about a little bit the bugs and how they're everywhere, and what do bacteria and microbes naturally do for human health? And can we repurpose these same microbes from native microbiomes for therapeutics? And then we'll talk a little bit more about how bacteria are the ideal chassis or host for these therapeutics. And then we'll talk about what they can actually do, what different illnesses can be treated with bacteria, and then a really quick discussion of some of the challenges that these therapeutics face. And so, yeah, let's get straight into the glossary. We're going to define some terms. So first, I wanted to talk a little bit about prebiotics. We'll basically define the term and then tell you if they're alive or not. And then I have a quick activity for all of you. And so first with prebiotics, they're basically the carbs or non-digestible food or insoluble fibers like fruits, veggies, whole grains that your body can't digest. So they basically go further along in your digestive tract, where there are foods to help, where they are then food to help the healthy bacteria grow. And so these are mostly whole foods like fruits and vegetables and other whole grains. And these are not alive. So these are just the food for the bacteria. Probiotics, on the other hand, are live bacteria and yeast, so your gut microbiome or gut microbes that support human digestion. So they can actually live in your body and help you digest the foods that you need. And those are alive. And then we have native microbiomes, and those are microbes that live and exist in every tissue in our bodies. And again, they're alive. And symbiotics are actually when we have prebiotics or the food and the bugs that actually eat those foods put together. So these are some ideas of like yogurt or kimchi, food for the bugs that are also fermented. And then last but not least, which is the main highlight of this presentation, are living bacterial therapeutics. And so there are a couple of different definitions, and the definition that I provided on the screen is actually the one that the US government has for living bacterial therapeutics, is that it contains live organisms, it's applicable to the prevention, treatment, or cure of a disease in human beings, and it's not a vaccine. And so these are living bacteria that are used for therapeutic purposes. And we can actually have probiotics become LBTs or living bacterial therapeutics if they're being delivered for therapy. And so I know these are a lot of terms, and I thought it would be easier if we actually spent some time looking at a diagram. And my first question for all of you is to figure out whether you can label the diagram with the correct terms. And we can pause here for a second. I'll give you guys a couple minutes to think about it, but I want you to assign the terms that we have on the left screen to each of the numbers that we have in the middle of the screen. And so we can pause and you can label the diagram with the correct terms in the chat if possible. We're seeing some different answers in the chat. Looking good. I'm gonna give it one more minute and then we can chat about the answers. Cool. So let's get into the actual labeling. So I saw a lot of things in the chat, and I just saw someone label number one as symbiotics, which is actually the correct answer because it combines both the prebiotics, which is number two, and the probiotics, which is also in your gut. And so number four, if the probiotics are delivered to the gut, they can be also within your human body. And so it's either probiotics or the native microbiome. And then the engineered living bacterial therapeutics are the ones that are actually doing this preventing of the different pathogens. So again, number one is synbiotics, where we're combining pre and probiotics. Number two is prebiotics because it's just the food. Number three is the living bacterial therapeutics because they're doing some function within the gut. And number four is the probiotics because they're in the gut and also in the can also be the native microbiome. Cool. So hopefully that makes sense to everyone. This is kind of why I wanted to throw it up on a diagram because I think it can be a little bit confusing without a picture. And so if anyone still has any questions about that, please let me know. And so with that, we'll get into the next part of our presentation. I can pause too for a second if anyone has a question. Cool. I guess we'll move on. So now we're going to talk a little bit about microbiomes. And so, as all of you probably know, the gut microbiome is very diverse. It's a combination of so many different organisms that participate in helping you digest your food. And this is actually on the magnitude of trillions of microorganisms that populate your intestines, like bacteria and viruses and fungi, including a bunch of different yeasts. And there's a lot of different dominant and rare species, but also some of these different transient species that can colonize your gut based on the things that you're eating or the environment that you're in. And I bet some of you are also recognizing some familiar names on the screen, like E. coli, which is a powerhouse in terms of synthetic biology, and Bacteroides species that have been commonly used to engineer different native functions into. And so we'll talk a little bit more about that later, but these bugs that we engineer are also very present in the gut microbiome. And so the gut microbiome actually changes as we get older and grow up. And each one of us has a unique microbiota, kind of like a fingerprint. And so when you're born, um, you basically get the microbiome that your mom has delivered immediately to you. And your microbiota will then change very dramatically from the ages 0 to 3. And it takes around 3 years for the gut microbiota to build up diversity and then stabilize. And so after that, when you're an adult, it's pretty stable. Um, and during adulthood, the composition or the different members within the microbiome are pretty much the same. And as you get older, the microbiota will then start losing its diversity, and it undergoes a lot of different changes in relation to it's both the host having an effect on the microbiome and the microbiome having an effect on the host. And so what does our microbiome actually do for us? And so the gut microbiome is actually very important to our overall health. And there's a couple of different functions on the screen, but I want to really point out 2 of them. And the gut microbiome first is really important for the first reason that we touched about really briefly at the beginning, because it promotes digestion by facilitating the absorption of nutrients. And so it helps you pick up sugars, amino acids, vitamins into your intestinal cells through the fermentation of a small fraction of food. And as the food goes further down in the gut, there are different bugs that colonize different portions of our gut and will process and break down that food accordingly. And these fermentation processes basically produce gas and numerous of different metabolites, including short-chain fatty acids, which is the real fuel for your cells. And the other function that I really want to point out is this defensive role or building a barrier. The gut microbiome is really important in building this barrier against pathogens and toxins and really helps out the immune system. And so even beyond that, the bacteria actually release these antimicrobial molecules that can kill the bad bacteria or the pathogens. And others even stimulate the production of this mucus or lining that sits on the edges of the gut, also providing a physical barrier against things getting across and into your body. And so with that, I have a question for y'all. What is not a function of the gut microbiome? And we can again take answers in the chat. And so we have A, promote digestion, B, stimulate mucus production, C, support the growth of pathogenic bacteria, and D, form a key barrier against pathogenic bacteria and toxins. I'll give it another second, but it looks like everyone's in agreement with the answer. So yeah, the answer is C. It does not support the growth of pathogenic bacteria. It actually sometimes kills off the pathogenic bacteria, which is quite interesting. And so beyond just our
hello?
gut, yeah.
I'm sorry, I'll take you a little bit back, just a little.
Great.
on the previous slide, 2 slides back. Huh?
Yeah.
Maybe, yeah,
This
no, the other one. Next,
This
behind. No, the one behind, please.
Okay, this one?
No, go back again.
This one?
Yeah, this one.
Okay, cool.
Yeah, thank you. I've just been thinking about it, and that's why I've brought you back. Huh?
Yeah,
You
totally
talk
fine.
about Escherichia coli being rare in the gut microbiome. And I thought otherwise because Escherichia coli is very prevalent in the large intestines and is very critical in the formation of things like vitamin K? So how does it become rare?
Yeah, so this is just, I think, data based on abundance. So it might be present in the microbiome, but it might not just be as present as some of these other species, if that makes sense.
Okay.
And so,
All
like,
right.
the
Okay.
majority of the microbiome is actually facilitated through those dominant species on the left. And so there are those other bugs that are important, exactly what you're saying, that do specific functions like make vitamin K for us, but they're just in the gut in lower abundance.
Okay, got it. Thank you.
Yeah, no worries. Great question. Okay, awesome. So let's move on to this slide. If there's no more questions, I can pause again, just in case. Cool. So yeah, microbiomes exist everywhere beyond just the gut. And like we've been talking about a little bit, there are many different factors that can affect microbiome composition, like your genetics, the environment, the diet that you're eating, the lifestyle, different hormones, and industry, meaning like what types of interactions are you having with both your environment and the things that are coming to you through the industrial landscape. And so all of these different things can have a really impactful effect on your microbiome. And the microbiome works in harmony with a bunch of different various organs in the body and aids in the proper functioning of a human being. And what you can see on the right-hand side of this figure are just some examples of that. Like, for example, the nose, you need that mucus production that we were talking about earlier. Or I like thinking about the skin, because on your skin there's its own microbiome and it's preventing you from interacting with all of the different bad bugs that are actually in our environment. And it also, the bacteria that are on your skin are actually giving you the scent, like when you sweat. Those are actually your skin microbiome releasing different volatile compounds. And so, yeah, there's a lot of reasons why we need these microbiomes, and I don't need to continue convincing you that they're important. But beyond microbiomes existing in every tissue, they actually also exist in solid tumors. And so in our lab, we like thinking of this tumor environment or tumor microenvironment as actually a microbe environment where there are microbes that are actually in the tumor environment. And why that is actually the natural properties of tumor tissues make them really great environments for supporting growth. They're full of different nutrients, and there's chemoattractants actually that pull in and grab the circulating bacteria inside the tumor mass. And so basically these bacteria are attracted to these chemical signals and then stay within the tumor mass, primarily because inside of the tumor, they're actually protected from the host immune system. So there's been a lot of different studies where immune cells basically can't get into the depths of these large masses of cells in the tumors, but because bacteria are so small, they can actually squeak through the leaky vasculature or the spaces in between the cells and get inside the tumor mass and proliferate and grow in there. And so there's been a lot of studies recently, especially about this tumor microbe environment. And if they already exist and thrive in these environments, can we use these bacteria that are already present as living, breathing bacterial therapeutics to get as a delivery mechanism, basically, to get it inside the tumor masses. And so with that, we're going to talk a little bit more about what makes a good therapeutic in regards to being a living therapeutic. And how can we actually translate this to living bacterial therapeutics? And so what are the requirements for a perfect therapy? So when we're thinking about a therapy, what if we had a dream list, what would it be? So this on the left is like a robot factory, and what we would like it to have is have it be extremely targeted to specific tissues. We want to make sure that it's not just being expressed all over the human body and having an effect. We want it very localized to a tumor or a specific type of tissue. We want it to self-propel towards a tissue. So basically similar to what we were saying, we want it to be able to get into the regions that are inaccessible to the immune system or other passive therapies that diffuse into the solid masses. Because right now it's really hard to get inside the big clump of cells. And in addition to that, we want it to be able to sense the local environment. And so we want it to be responsive to make decisions about where and when these drugs are administered. And that responsiveness will then would allow us to precisely control the location and the timing of the delivery of some of these different cytotoxic molecules or therapeutically relevant chemicals. And we also would like the cell to produce externally detectable signals so that we can understand the current state of the tumor, the success of localizing the bacteria to this region, and also the efficacy of the treatment. Like, how well is our therapy doing at these cells? And so my question to all of you is, what are ways that bacteria already perform some of these functions? And you can think about this in respect to what we were looking at before with the microbiome or some native functions that microbes have. And you can just throw those in the chat again. And so how do microbes, based on your understanding of this course already, and some of the other lectures, already do some of these different things, either natively or through engineered function? Seeing some good answers. Efflux pumps and quorum sensing are definitely important. Yeah, someone said that they're following the attractive signals released by tumor cells. Yeah, we just talked about that. That's a good one. Biofilm formation is another good one. Chemotaxis, all of these are great. Cool. Let's keep going. So bacteria have actually a lot of biological mechanisms to perform a lot of these different functions. And so they have genetic machinery to produce a bunch of these different compounds that can be then released to the cancer cells and kill off the cancer cells. They can have specific promoter regions to respond to different molecular signals. So only turning on when the molecular signal is present. They have chemotaxis receptors, which is something that someone mentioned in the as a response to the previous question, which basically allows them to motorize their flagella and move towards the environments that they're responding to. And again, they have all of this machinery to produce all of these important molecules to then kill off the cancer. And so, wow, bacteria are really great for the sense and response. And a lot of people are really excited about this field, and it's definitely an up and coming area of synthetic biology. And so I really wanted to talk a little bit more about how bacteria transport themselves and migrate, and why it's different than the traditional chemotherapy approaches. And so, yeah, in the chat, someone asked, given the difficulty in accessing the target, sorry, is there a potential for application of nanotechnology in bacterial engineering and synthetic biology? Really good question. I actually have some slides about that at the end, and so we will definitely get to that later. And the next question is, how would the engineered living bacterial therapeutic be delivered to the tumor environment? Fantastic question. We will also get to that. But on this slide, what you're seeing is injections, and that's pretty standard right now for how we are delivering bacterial therapeutics to the body. So through an injection is pretty standard. There are also some other research groups that are focusing on delivering the bacteria orally. So the patient would take a pill that would have the bacteria. And so they have done some studies about how the bacteria actually home to, like, from the gut to the solid tumors. And so I can forward you along some papers if you're interested in that. But yeah, back to the slide. So what we're seeing here is a blue mass of tumor cells, which you can see in the body right here. And we're delivering this hypothetical patient both an active bacterial therapy and a passive chemotherapy. So the active bacterial therapy can get through this big mass into the center of the mass and actually deliver different therapeutics inside of the mass, the solid tumor. And the chemotherapy is very dependent on how close the blood vessels are, and also its passive diffusion into the solid tumor mass. And so bacteria can get, again, further into these proximal regions. So we see more of the bacteria in these closer regions than we see the passive chemotherapy. And at these distal regions, they're already very present. And then so when they're injected, like what we talked about before, they're going to accumulate in the tumors and migrate away from the vasculature or the whole body. And this is really beneficial to us when we're thinking about targeting, because if they were in the whole body, also known as like systemic administration, they'd be actively cleared from the immune system. So your immune system is really good at picking up and destroying foreign contaminants. And so if we were delivering it all over, it would actually clear out the bacteria from the environment. And so they hide from the immune system inside of this huge cluster of cells, which is exactly where we want them to be to deliver the therapeutics. And so to talk a little bit more about the key components of engineered living bacterial therapeutics, and now that we know a little bit more about what makes a perfect therapy, we can get a little bit more into what elements are there in engineered bacteria. And so we need to have biomarkers that we are activating or responding to, and often these are small molecules that are either made by hosts or bacteria, secondary metabolites, physiological conditions like oxygen and temperature, which are unique sometimes to different environments. They can also respond to the gut microbiome itself, as all of those bugs have their own biomarkers or indications that they're present. And so once we have that signal received from some of these different things that can trigger our therapeutic, we can use promoters to have very specific control over bacteria in a location. And so those promoters can be tuned so that we can have different expressions states. So basically, if you have a low expression promoter, we'll have less of the drug induced. If we have a high expression promoter, we'll have more of the drug produced. And then we can the things that we can express can also change. So we can express different proteins, peptides, enzymes, metabolites, all of the same things that you've heard everyone talk about in previous lectures, we can express in these same bugs. And then we have to find a way to actually release and get these therapeutics outside of the cell. So we can do that either through secretion, so like forcing the therapeutic to go through different effectors and get through the membranes of the cell it's currently in. We can also do cell lysis, which actually kills off the cell. So it rips apart the cell open. And then once the therapeutic is out and in the environment, there's a bunch of different things that it can do in regards to disease targeting. It can kill cancer, have modulation of your immune cells. It can heal that mucus barrier that we were talking about before. There's also antimicrobial activity and different restoration of different components within the microbiome. And so there's a lot of things that you can change and tune using synthetic biology tools to have engineered function in these cells. And so to sense the specific health environments, again, we can use these engineered promoters. And I wanted to give you some ideas of what kinds of things you can sense. So you can sense oxygen conditions, pH, glucose, radiation, and some health-specific environments are actually specific to these molecules. Like, for example, cancer environments are often hypoxic and have low pH. And so if you have promoters that can actually sense this, then you can respond to the environment and produce different molecules of interest. And so what kinds of things can we respond with, and what kinds of things can we make? And so we can make different reporters, we can make cytokines to turn on the immune system, we can do have inducers of apoptosis lyse open the cancer cells themselves. And so there's a lot of different things that we can do with this set. And so now that we've talked about all of this, my question to all of you is, you have to tell me if this statement is true or false. Engineered living bacterial therapies can only be used as cancer therapies. We've talked a lot about cancer. Is that the only thing that we can talk about in regards to using bacterial therapies? False is correct. So yeah, the answer is that obviously no, we cannot. There are a lot of other things that we can do with these bacterial therapies. And now we're going to get into them. And I'll pause right here for a second and see if anyone has any questions, because we did go through a lot of content.
I'm just getting a little concerned. Hayo, can you hear me?
Yes, yeah.
Hello?
Hi.
Okay. Just in the previous, hi to you. In the previous slides, one of the possibilities of delivery and what would then eventually happen to the cell that is delivering it, the bacterium, which is the LBT then, would be lysis. And, in the mind of, say, the people in the health sector, or even we as synthetic biologists, wouldn't that be one of the issues of concern? Because the material within bacteria, after it dies, becomes unwanted in the body and it accumulates, and that's probably giving the liver a lot of work to do. Can that also be an issue? Yes, please go ahead.
Yeah, that's totally a really great concern. I think in the past, people have done mice studies to ensure that the liver is still functioning after having to clear some of this increase in microbe waste, I guess. But the fun part of this all is that your body is constantly doing this with microbes that already exist and you interacting with them. And so it has a lot of mechanisms to do this. And it, as long as the dosage isn't too high, your body is completely fine with clearing out additional microbes through whatever mechanism, the liver or whatever. And I think we'll talk a little bit more about cell lysis and how it works, but it's kind of cyclical. So you'll have periods of lysis, and then using whatever's remaining, you'll grow up a new set off of there. And so it won't be like a constant barragement of the immune system, if that makes sense. At least with the lysis system.
Okay, thank you. Thank
Yeah.
you for that.
Yeah, of course. And then Nelson has a question on how specific is cell lysis in respect to self and non-self cells. Very good question. We will actually have some slides on lysis specifically at the end. And so I'm gonna keep moving forward just so that we can get there. That sounds good, Nelson. Awesome. So there's a lot of potential uses for living bacterial therapeutics. And I wanted to really quickly show this slide because in 2010, there were about 30-40 papers in bacterial therapeutics. And now in 2020/2025, there are so many more. It's on the scale of like hundreds and thousands. There's a lot of people that are excited about this and engineering these organisms to be bacterial therapeutics, including our lab. And so there's a lot of room for exploration in this area. And so now we're going to talk a little bit about a historical example about Colles toxin, some modalities for cancer therapies, and some examples of lysis circuits. And then a whole nother application in regards to a metabolic disorder and how a company in the US is trying to fix that metabolic disorder, which is not cancer related. Yeah, and so if you've ever taken a microbiology class, they probably talk about Colles toxins, but William Colles was in the 19th century came across the description of a patient with an inoperable sarcoma. Sarcomas are bone cancers whose tumor basically completely vanished after developing a type of skin infection. And so what William Colles did is he took these toxins, or this bacteria, which was heat killed from Streptococcus pyogenes and Serratia marcescens, and then delivered it to a sarcoma patient and found that it activates the immune system, and that patient actually survives and is a healthy patient. And later on, we figured out that this was related to IL-12 production. But this was basically the first famous cancer immunotherapy, and it's all from bacteria and bugs, even though that they're dead. And so this was a really horrible, unsafe way to do science, but really advanced fundamental understanding of cancer. And now is some of these core elements are used in different immunotherapies to this day. And so beyond cancer, what can we do with engineered living bacterial therapeutics? We can use it to target inflammatory and autoimmune diseases, cancer, obviously, target infections and different pathogens, and then also metabolism and cell reprogramming. And so we have one example on cancer and another example on metabolism. And the truth is, like on that the papers diagram, a lot of these therapies have focused on cancer, and it's primarily because it's really prevalent. It's like a disease that affects a lot of people, and there's a lot of funding in the US for this. And so I wanted to talk a little bit about what advances have come in the last couple of years in terms of cancer related bacterial therapeutics. And so we were talking a little bit earlier about targeting tumor environments, and there's two different ways that we can do this. We can either do intracellular delivery, where we have direct interaction with the tumor cells, or we can do extracellular delivery, which just means that it's released into the local environment. And within that, there's a bunch of different things that we can do to actually disrupt the tumor cells. And I don't think that we have enough time to talk about all of the different things. And so I'm going to focus really on this concept of lysis because it's very prevalent in the literature, and I think something that to me is quite interesting. And so we're going to talk about this first example through the lens of a company that was actually founded in building commercial engineered living bacterial therapeutics and lysis circuits. And so I'm going to first walk you through how this works, and then we'll watch a fun video. But basically, the bacteria will have long-term infiltration inside of those solid tumors, exactly in the same ways that I told you before. And what happens actually is when there's a lot of these microbes present, they use quorum sensing, which is something that we've talked a little bit about, to turn on the expression of genes that will then lyse the whole population. So basically, they're making all of these small molecules that are called AHLs, and these molecules interact with different promoters and turn on the lysis circuit, or the lysing of these bacterial cells. And that allows them to have very precise self-triggering expression for really high dose local delivery. And bacterial lysis basically triggers the delivery of these payloads and this immune system primary via different stimulatory ligands that are then delivered into the local environment. And then basically what you can see even through here is that there are a couple survivors, and the lysis basically kills most of the population, but not all of the population, and then this cycle continues. And so you have growth, then overgrowth, which has a lot of these AHLs released in the environment, and then those trigger bacterial lysis and delivery of your payloads. And then with whatever survivors are remaining, we repeat the process. So hopefully that made sense. Now we can watch a fun video while you guys think about this concept. It's pretty cool to see this process happen visually with GFP. And so what you're seeing is the cells proliferate, and then they release the cargo, and then they start over. And it's pretty cool. And there's a lot of videos of this on YouTube. And when I was learning first about microbiology, like 10 years ago or whatever it was, this is one of the videos that actually got me into it, got me into synthetic biology and learning more about microbiomes and microbes in general. I just think that it's very cool to see how all of this works. Yeah. Cool. Okay, if there's no more questions, we'll proceed. And so what can we do with these engineered living bacteria? The company's name was GeneCirc. They're housed at UC. They came out of a lab from UC San Diego, and I think they're still doing cool things with lysis circuits. Yeah. So what can we do with engineered living bacterial therapeutics? This next example will be from a recent startup, Synlogic. They actually shut their doors earlier this year, but they made really monumental strides in getting some of these bacterial therapeutics approved by the US government. And so what they're trying to do is basically treat this condition called phenylketonuria, or PKU, which is way easier to say, so I'm going to be saying it like that from now on. And they basically, in PKU, if you standardly absorb amino acids from different proteins that you eat. And if you're healthy, you have this enzyme called phenylalanine hydroxylase, or PAH, that converts phenylalanine into tyrosine, and then your body uses that tyrosine. But in PKU, they actually have an impaired PAH, which means that they have a lot of this phenylalanine build up inside of their body, and to toxic levels. And so what Synlogic did is they built an engineered probiotic bacteria with different components of synthetic genetic circuits, and actually made and expressed this compound PAL and this enzyme PAL and this other enzyme LAD to basically break down the phenylalanine into things that your body can actually use. And so I don't have to go too much into the details, but they've done a lot of optimization of these circuits to respond and produce different enzymes for your body, for PKU patients to survive. Yeah. And so my question to all of you is, what are ways to engineer genomic stability in bacteria, and how could we do it with this SynLogic strain? And you guys can throw that in the chat. So like based again on your experiences in engineering biology so far, I want you guys to give me some ideas of how we can engineer genomic stability into bacteria. It's a hard question, I know. Nelson, are you asking like what stability is? Okay. Okay, sounds good. Rec aging is interesting. That's an interesting idea. Cool. I'm gonna move on. I'll tell you what they ended up doing. So they basically their in the genome of this E. coli Nissle, what they did is they put multiple copies into the genome of each gene that they were actually expressing. So they put like 3 or 4 copies of this STLA gene, which expresses one of those enzymes I was talking about, and then put the other enzyme under a different promoter also in the genome. So the answer to this is genomic integration for SynLogic, and also multiple copies of each of the gene integrated into the genome of E. coli Nissle. So that's what they ended up doing. Yeah, and they did a lot of different work optimizing these genetic parts using high expression landing pads, basically meaning at those sites they have better expression of all of the different genes that they want and better maintenance of the genes that they want to keep inside of the bacteria genomically integrated. And so, yeah, and so what can we do with engineered living bacterial therapeutics and their interactions with other technologies? This was a question that was asked earlier, and this goes beyond just living cells, but can also include living cells. So bacteria can be used together with both living and non-living technologies for improved diagnostic and therapeutic outcomes. And so there was a lot of discussion in literature about how we can use different nanotechnologies like nanoparticles and different imaging techniques to visualize what was actually happening at these sites. And so you can use both living things like microbial consortia, where you can have interactions between multiple microbes producing the thing that you want, and also things like the nanotechnology side to further improve diagnostics and therapeutic outcomes. And so really quickly, we're almost at time, so I'm going to blast through the rest of this. There are some challenges that are related to actually implementing these technologies. So there's a lot more challenges that are related to using these therapies in vivo. And depending on how you're administering the bacterial therapy, it'll have different barriers to effective delivery. And so when you're injecting or using intratumoral injection, you can get systemic immune response and toxicity owing to the presence of some exact response of your immune system to the bacteria. There can also be colonization outside of where you want it to happen if those promoters are related to other environments that are also present in other tissues. And so you have to be very specific about how you trigger production of your cytotoxic compounds. And so often what people will do is they'll actually have two promoters so that you have to have both promoters on like in the right environment. So like an AND gate, both promoters on in order to express whatever downstream therapeutic you would like to express. And like I was mentioning before, there's a lot of ways that your body naturally degrades and clears bacteria on its way to the cancer cells. And so the therapeutic has to be able to get past that. And once it's actually at that site, it's really important to control the colonization of the bacteria, transport the payload to the proper location, and also make sure that there's an actual therapeutic effect against your immune cells. And so, yeah, this is a very quick explanation of some of the challenges that we face with living bacterial therapeutics. And that's basically it for today. So a summary of today's lecture. There are many terms related to probiotics and engineered living bacterial therapeutics. Microbiomes are everywhere in our bodies and perform important functions to maintain our health. Bacteria are the perfect host for living therapeutics, as we can use a lot of different synthetic biology tools to build different therapeutic functions into bacteria. We can treat a lot of different conditions with bacteria. Even though this presentation really focused on cancer, we can treat some of those metabolic disorders and inflammation. There have been many key developments in synthetic biology that have improved our ability to use engineered living bacterial therapeutics, but there are still key challenges to using these in cells and inside your body. And yeah, if you have any questions, you can always Slack me on the SynBio4All Slack page. I also put my email and my X up here if you want to connect in other ways. And hopefully this topic gave you another source of inspiration for your projects, especially when you're trying to come up with different ways that we can use synthetic biology. I think the health sphere and engineered bacterial therapeutics have a lot of different applications to explore. Yeah, and so that's all. Yeah, and if you guys have any more questions, we can definitely answer those now too. So Nelson is asking, how long will the therapeutic process take? I assume it isn't instant. Yes, that is true. It really, because of all of the tools that we have, it's pretty controllable with how you actually how fast the response is. But you have to remember that the bug has to get to the environment first to then be able to release its payload. And so that can take some time. I think standardly with the SynLogic experiments, I think that they saw it happen across at least the scale of like 8 to 24 hours. That sounds right. Yeah, I would have to look that up. I don't know the definitive time frame. I think it very much changes based on the therapeutic. Yeah. Cool. Awesome. So there's how do we manage the dose aspects? The dose aspects. Can you clarify what you mean by that? Do you mean like how much of the therapeutic is being produced? I can come back while you. Yeah, how much of the drug the patient will receive. Yeah, so all of that will very much depend on what promoters the therapeutic genes are under, and also um, basically what the expression cassette looks like. When we take things from a plasmid to the genome for stability, we actually we lose copy number, right? Like we lose the number of copies that we have inside of the cell because now it's genomically integrated. And so there's a lot of tuning that needs to happen with making sure that your expression vectors are actually producing a compound that is viable as a therapeutic. And so I think a lot of people have done a lot of extensive characterization of their chosen therapeutic. It's just very specific to whatever you're trying to deliver to make sure that it's enough for the disease. Okay. Okay. You gave an example. Yeah, so those are some, I'm gonna mute whoever that is. Sorry. Okay, so you gave an example of two companies, GeneCirc and SynLogic. Are those methods being applied in real life, or are they just in the early development stages? So a lot of these companies are starting now or have gone to a couple phases of trials, but they are definitely in the infant stages of their journeys as therapeutics. There's also this, people have also developed T cells, which is an immune cell for as a therapeutic called, I bet you guys have heard of this, like chimeric antigen receptor T cells or CAR T cells. CAR T cells have had a lot more promise, and there's actually, I think, 6 or 7 that are actively available on the market right now. So those are living therapeutics that are not bacteria, but they are human cells and can be and are being used actively as therapeutics. Yeah, but bacteria are still in the infant stages. There's some companies that have closed down recently that were probably at the forefront. It just, there's a lot of testing that needs to be done, and you need a lot of money to do that. And so, yeah, it's kind of unfortunate, but hopefully there'll be more as time goes on. Yeah. Okay, what typically happens to the remaining bacteria once the patient is healed and they are no longer required? Yeah, so the bacteria, so in order for the bacteria to proliferate in those environments, it requires like a constant supply of either the injection or the oral delivery mechanisms. This is actually a really good question because your immune system eventually does get to the bacteria, and it's eventually cleared from the body. And so as long as you are continuously taking the living bacterial therapeutic, you will have it present and working. But as soon as you stop, they've shown that it'll clear from the human body. To my understanding, yeah, I think that's something that I would definitely go back and look up. Yeah. Okay. How can you program one bacterium to go after another pathogen? Yeah. So there are specific markers for pathogens, just like there are specific markers for cancer cells. And so often they're referred to as PAMPs, or pathogen-associated molecular patterns. And often those are either extracellularly or like on the outside of cells, displayed on the cells. And so if the bacteria can recognize that PAMP exists, then it can then turn on a circuit and kill off the pathogen. And so there are some companies and some research labs that are interested in PAMPs and using those as a biomarker of disease state, but also as a therapeutic target itself. Yeah, okay. When applied in real life, won't there be an impact on other bacteria?
Can I ask a question?
Oh, yeah, absolutely. Yeah, go ahead.
Okay, I just wanted to, I'm still flabbergasted with the use of LBTs. There is one of our participants here who has mentioned something about self and non-self cells.
Yeah.
With regard to what the body would recognize and what it doesn't. Because at the rudimentary level, we know that anything that is foreign, like a bacterium, which is a protein, will be fought against by the immune system.
Mm-hmm.
I don't know whether. So does it mean that you also induce or induce those cells, or are we just looking at them as probiotics and therefore they are recognized by the body? Because we are talking about something that gets into the bloodstream.
Yeah. So if it's in the bloodstream and recognized by the immune system, it will be cleared from the body, right? This idea of self and non-self is like a training mechanism within your immune system. So as it perceives different threats, it will then recognize it as a threat and then clear it from the body. I don't actually know the mechanism of how it recognizes or if it recognizes specific bacteria, like if there are specific commensal organisms or like the ones that are not harming us, that kind of live in symbiosis with us, not that don't affect, that aren't killed off by the immune system. And another set, which is like our engineered therapeutics or pathogens and how the body actually recognizes those. I actually don't know off of the top of my head. I think that's a good question. Sorry if that doesn't answer your question. I apologize.
No, we will still dig and find out more. Thank you.
Yeah, of course. What is the difference between the mechanisms used by LBTs and phage therapy? I think they both interact with the cancer cells in similar ways. So you can make similar things in them, but I bet that the way that they recognize the environments that they're in and the expression vectors and processes are quite different between the two. My background is definitely more bacteria and more bacterial tools like ice. But yeah, this is definitely phage is definitely not my area of expertise. Yeah. Cool. Could you explain the notion of probiotics a little bit more? Also, do they occur naturally? What is the link difference between probiotics and engineered probiotics? Good question. We can jump to the slides at the beginning. So you can consume probiotics, and those probiotics are bugs that have already existed standardly in the gut microbiome. An engineered probiotic standardly is not the wild type version of that bug. It is with additional components within it. So an LBT can be just a plain probiotic, but it could also be something that has been engineered to have a specific function in the gut. Hopefully that answered your question. Cool. People are chatting about phage, fantastic. Probiotics shouldn't be able to initiate immune reaction. Correct. Nelson's talking about a paper he's writing. That's cool. That's fun. Love. Good job. Are there any other questions? You guys are so engaging. This was very fun. And yeah, again, you guys can contact me later with more questions if you have them, if you don't want to ask them here.
Hey, Ava, I don't know if you have already talked about it, though I didn't hear it. Maybe could you please clarify on the patient safety when you're using LOBTs?
Yeah, you're asking about safety when using like engineered living bacterial therapeutics. Yeah. So
Yeah.
there's a whole field and branch of synthetic biology that also touches this concept. And the field is like called biocontainment. And basically through things like engineering genomic stability and making the bugs actually oxytrophic. What that means is like they need this DAP-A compound in order to survive. You can engineer uh, specificity to and containment of the strain only when this DAP is provided. And so there's a lot of different research that is being conducted in biocontainment, and both with like non-standard amino acids to like make sure that this bug can't survive outside of this environment where it's supplemented with all of these extra things. And then other issues with stability and making sure that things are only surviving where we want them to survive. I think there's some cool stuff that's happening right now with genetic kill switches. And so basically, when the bug escapes a particular environment, it will die and only proliferate in the environment that is desired. And so it's kind of similar to a lysis circuit in that way, where it's like, oh, if I leave this space, I die. Instead of like with the lysis circuits, like if I sense these molecules, then I die. Yeah. I don't know. Does that answer your question? Cool. Okay, another question. Could you talk about dysbiosis in relation to the gut microbiome and probiotics? Probably not, because this is not my field. Um, yeah, I think these strategies are very, very cutting edge. To my understanding of how something like that would work in terms of like the synthetic biology aspect, you could sense dysbiosis, which would maybe mean production of specific compounds or the presence of specific stress responses, and then respond to the dysbiosis by providing something that would reduce inflammation or reduce some of the factors that are leading to dysbiosis. That's basically what I understand the technologies to be. But yeah, I think this kind of stuff is definitely very cutting edge. A lot of people are also, I think there's a larger front pushing forward in bacterial therapeutics, at least in the States, towards using probiotics in their wild type form. So like without the without engineering any additional function and just delivering either a consortia of organisms, so multiple organisms together into a host, or just a single organism that maybe is missing from that environment and they're supplementing it in. And so yeah, that's basically some things that people are pursuing within the gut dysbiosis space. Yeah. Yeah. Cool. Yeah, I think I agree with Emma. Let's move this over to Slack if you guys have any additional questions, but it was really fun talking to all of you. And yeah, good luck with your projects. I'll be around if you have questions. Yeah.
Okay, thank you so much, Ava, for such an engaging session. And really, it was a very nice lecture, and I hope all students have appreciated the lecture. So I would like to encourage the SynBio4All Africa students. For those ones who have not yet got groups, working groups for your project, please, we shall group you as SynBio4All. We shall group you. You can just fill the form. The form is there. It's circulating. You can fill the form, and we shall help to group you. And you can also start submitting the project ideas which you have, that they can be, our instructors can go through them and look at a project that can really be done within a short period of time, and it can be beneficial to you. Also, I would like to appreciate whoever attended today's lecture and whoever attended the previous SynBio4All Africa webinar, the guest speaker event whereby we had Yoko from IGEM. And very soon we shall have the June webinar, and that will be on the 21st of June, whereby we shall host Alex Diesel. Alex Diesel is the founder of Project. You can go and make research about Project. It's a student competition which happens online. So your projects can also really be fit for the Project competition of this year. So you can check on Project and see what it means. Otherwise, thank you so much for attending today's lecture, and I would like to give back to Ava to give her final remarks as we conclude today's class. Thank you so much.
Yeah, thanks, guys. Really enjoyed this. Yeah, you guys are so engaging. That was so fun. Yeah, nice chatting. Again, if you need anything, I'm on the Slack.
Okay, thank you so much. I think Ava is done with her final remarks, and I wish you the best of whatever you're doing. And we meet again on Wednesday. Don't forget to work on your projects and to get teams. Thank you so much.